Hello and welcome to the new session of the lecture series, and today's topic is cell cycle and cell division. And today we'll be discussing and solving the NCERT based multiple choice questions. Okay, so let's start with the first question. So here they are saying just the G0 state. G0 state means the quiescent state or the inactive stage of the cells in eukaryotic cell cycle denotes, okay, now we already know interphase, okay, interphase is divided into G1 S phase and then the G2 phase, and then we have the mitotic or the M phase. So they are asking about the G0 phase. So sometimes what happens, some of the cells such as cardiac cell, which are heart cells, the skeletal muscle cells, some lymphocytes, alright, they exit the G1 phase and they enter the G0 phase. And G0 phase is an inactive phase. Okay. It is an inactive phase, but the cell is metabolically active. Okay. But the cell is metabolically active, but it is not proliferating, means it is not dividing. Alright. So options are that, let's read the options. First is the checkpoint before entering the next phase. Alright. Second is the pausing in the middle of the cycle to cope up with the temporary delay. Third, death of the cell or exit of the cell from the cell cycle. So we know the cells sometimes exit from the G1 phase of the cell cycle and they enter the G0 phase. So what should be the answer? Option D should be the answer. Alright. Option D is the answer. Now coming to the next question. During which phase of the cell cycle amount of DNA in the cell remains at 4C? Okay. You can circle this. They are asking about 4C. If the initial amount is denoted as 2C. Alright. So again, we are talking about interphase here, G1, S and G2 phase, which are the subphases of interphase. Okay. So if the organism is diploid, means it contains two sets of chromosomes. Alright. In S phase, what happens? DNA replication takes place. Okay, here DNA replication or DNA synthesis takes place. Here initially DNA content is denoted as 2C, but after the S phase, what happens? DNA content will be 4C. But the chromosome number will still remain 2n, means the organism will be diploid only. Okay, but the DNA content has doubled due to DNA replication during the S phase. Alright. So options are G0 and G1, G1 and S, only G2, and G2 and M. So what should be the answer? DNA concentration at G2 will be 4C, but the chromosome number will be 2n. So the answer should be only G2. Alright. Now coming to the third question. Here they are asking which of the following phase follows S and G2 phase of the interphase. Alright. So again, let's talk about the cell cycle. cell cycle is divided into interphase, and then we have the mitotic phase or the M phase. Alright, so we have the G1, S, G2 and the M phase. M phase is further divided into prophase. First phase is the prophase. Alright, M phase you can say is divided into first karyokinesis. Okay, karyokinesis means nuclear division. Okay, and after karyokinesis is cytokinesis, means cytoplasm division. Okay, karyokinesis is nuclear division, division of nucleus, whereas cytokinesis is cytoplasmic division. So karyokinesis is further divided into following phases: prophase, then we have the metaphase. After metaphase is anaphase, and then we have the telophase. And after telophase, cells undergo cytokinesis. So here what should be the answer? Answer should be prophase. Okay. After the G2 phase, we have the M phase, and the first phase in the M phase is the prophase. Now the next question is, in mitosis they are asking, nucleolus and the nuclear membrane disappear at which stage? Interphase, prophase, metaphase and telophase. Alright, so interphase you already know, okay, about G1, S and G2 phase. Prophase, what happens? At the end of prophase during mitosis, nucleolus, okay, and the nuclear membrane, they disappear. Nucleolus and the nuclear membrane disappear, or you can say nuclear envelope disappear. You have already got the answer now. Metaphase, what happens? Chromosomes are aligned at the equator. Chromosomes are aligned at the equatorial plate, also known as the metaphase plate. Equatorial plate is also known as the metaphase plate. Okay. Telophase, what happens? It is the reverse of prophase. Here what happens? Nuclear membrane, nucleolus, everything, they reappear, as well as the cytoplasmic organelles also reappear. Okay, so what should be the answer? Answer should be prophase. During prophase, nucleolus and the nuclear membrane, they disappear. Now next question is, best stage to observe sharp, okay, shape, size and number of the chromosome is, okay, which is the best stage to observe the shape, size and the number of the chromosome? Alright, is it interphase, is it metaphase, or it is prophase, or it is telophase? Okay, these are the options. So what happens? Interphase, you already know, cell growth is taking place. Okay, cell growth is taking place, cell is metabolically active. Okay, final preparation for the cell division is taking place. And what else is happening? DNA synthesis is happening in the S phase. Metaphase was, and then after interphase we have the prophase, okay, where chromatin condensation is taking place. Chromatin condensation is taking place, and due to this condensation, or you can say due to this coiling of the chromatin, chromosomes are clearly visible. Okay, but at the metaphase stage, chromosomes are clearly visible. Okay, they are distinctly visible, and it is the best stage to observe the chromosome. Alright, during telophase what happens? Reverse of prophase, the chromatin is, or you can say the chromosome decondense. Okay, in prophase it was condensing, here it is decondense and they become indistinct. Or you can say they lose their individuality. So what should be the answer? Answer is metaphase. Now coming to the next question. Cell plate grows from wall to center, center to walls, one wall to other, or simultaneously. So they are talking about cell plate means they are talking about formation of the cell plate in the plants. They are talking about the plant cell. So cytokinesis, or you can say the mechanism of cytokinesis, is different in animals and plants. It takes place by the formation of cell plate in the plant cells. Okay, cell plate is seen in the plant cells. So what happens? If this is a cell plate is formed in the center of the cell, and this cell plate gradually grows outward till it meets the lateral walls. Okay, so a cell plate is formed in the center and move towards the walls. So what should be the answer? Answer is from the center to the walls. Now next question is, which of the following cellular structure always disappears during mitosis and meiosis? Alright, so options are plastid and the nuclear membrane, nucleolus and the nuclear membrane, endoplasmic reticulum and mitochondria, and endoplasmic reticulum and plasma membrane. Alright, so you know at the end of prophase, what happens? Nucleolus and the nuclear membrane, they disappear. Alright, they disappear. So what should be the answer? Answer should be nucleolus and the nuclear membrane. Now next question is, during mitosis, nuclear envelope, nucleolus begin to form and ER reappear at which stage? Alright, earlier question they were asking disappear, now they are asking reappear. So prophase, we know there is disappearance of nucleolus and nuclear membrane and the cytoplasmic organelles. Metaphase, chromosomes are lying at the equator. Anaphase, what is happening? There is separation of the chromatids during mitosis. Okay, there is separation of chromatids during mitosis. That's why it is known as equational division. And what happens during telophase, which is the reverse of prophase? Okay, all the above mentioned things, nuclear envelope, nucleolus and the cytoplasmic organelle, they reappear. So what should be the answer? Answer is the telophase stage. Now the next question is the microtubules, okay, of the mitotic spindle. So spindle fibers are made up of microtubules. Spindle fibers are made up of microtubules. Alright, so microtubules attach to a specialized structure in the centromere region of chromosome. Okay, now we know if this is a chromosome, this chromosome is, or the sister chromatids of the chromosomes are attached with the help of the centromere. Okay, this is the centromere, which contains two barrel-shaped, okay, these are barrel-shaped structures known as kinetochore. And these are the areas where the microtubules attach. Okay, microtubules attach at this region. Okay, so what should be the answer? Answer is kinetochore. Okay, so option D is the correct answer. Now coming to the next question, which is, which of the following is the result when karyokinesis is not followed by cytokinesis? Alright, the options are synaptonymal complex, syncytium, recombination nodules, and terminalization. Alright, so synaptonymal complex, you know, it is a protein lattice. Okay, it is a protein lattice, okay, which helps in the assembly of the homologous chromosome, okay, or which holds the two homologous chromosomes together. Holds the two homologous chromosomes together. Now, syncytium, what is syncytium? Syncytium is a multinucleate structure, okay, which is usually formed by the fusion of unicellular or uninucleate cells, okay, forming a multinucleate structure. Alright, and sometimes they are also formed by the nuclear division, but usually syncytium is formed by the fusion of the uninucleate cells. Alright, examples of syncytium can be, first, if we see in animal cell, muscle cells, or you can say muscle fiber is the example. So what muscle fibers are made up of? Muscle fibers are made up of by the fusion of many muscle cells. Okay, and this helps in the coordination and control of the muscles. Syncytium is also found in some of the fungus. Alright, now recombination nodules. So recombination nodules are the area where crossing over occurs, or you can say they are the site of crossing over. Where crossing over occurs. Terminalization, they are saying about terminalization of chiasmata. Okay, where the chiasmata move to the terminal position of the chromosome. So what should be the answer? Answer is syncytium. Now next question is, mitosis is the process by which eukaryotic cells, first option is, expose the genes for protein synthesis, become specialized in structure, they multiply, or they show genetic variability. So mitosis, you know, is a cell division process, okay, and it forms two equal size, equal shape, genetically equal cells, okay, in the end. So option 1 is incorrect. Expose the genes for the protein synthesis, this is incorrect. Become specialized in structure and function, this is also incorrect. Okay, the cells become specialized in structure and function when they mature. Okay, but mitosis is about cell division, it is not about the maturity of the cells. Multiply, alright, this is the correct statement about mitosis, because mitosis helps in the cell repair and growth. Okay, so the cells multiply using mitosis and lead to the cell repair and growth. Examples can be seen in meristem. Okay, meristem, they consist of meristematic cells. Okay, they can be root apical meristem. There is shoot apical meristem. The cells in these meristem grow with the help of mitosis. Alright. So now you already know the answer. Answer is C. Genetic variability again is incorrect because variability is caused by meiosis and not mitosis. Because in meiosis, recombination and crossing over is seen. Okay, recombination and crossing over leads to variation. Alright, so the correct answer is multiply. Now coming to the next question, here they are asking in animal cell, cytokinesis involves, first is separation of the sister chromatids, appearance of a furrow in plasma membrane, depolymerization of kinetochore microtubules, and formation of cell plate. So in animal cell, what happens? Suppose if this is a cell, in animal cell, cytokinesis or the division of the cytoplasm, how it takes? There is a formation of furrow in the plasma membrane. Okay, there is a formation of a furrow in the plasma membrane, and this furrow gradually moves inwards. Okay, this will gradually move inwards and ultimately form two cells. It will pinch the plasma membrane to an extent, forming two cells in the end. Alright. So, correct option is appearance of a furrow in the plasma membrane. One point here to note that the mechanism of cytokinesis in animals is known as furrow method. You can say furrowing, or you can say cleavage furrow method. Okay, this is known as cleavage furrow method. The next question is the major event that occurs during the anaphase, okay, of mitosis they are asking, which brings about the equal distribution of chromosomes. Alright, replication of the genetic material, splitting of the chromatid, splitting of the centromere, or condensation of chromatid. So what happens during the anaphase of mitosis? Centromere splits. There is splitting of the centromere, due to which there is segregation of chromatids. And due to this, there is an equal distribution of the chromosome in the daughter cell. And that's why mitosis is known as equational division. What is the correct answer? Splitting of the centromere. Now next question is, which of the following is responsible for the formation of new cell wall and also represents the middle lamella between the walls of the two adjacent cells? Now first discuss what is the middle lamella. Middle lamella, okay, is a cementing material, or you can say it cements the primary cell wall, okay, primary cell wall of the adjacent cells, okay. If these are the two adjacent cells, these two are the primary cell wall. Okay, so middle lamella is present here in this region. Okay, it cements the to primary wall, okay, of the adjacent cells. This denotes the middle lamella. Alright. So we know in plant cell, there is cell plate formation, okay. Cell inclusion is not correct, cell membrane is not correct, okay, none of these is also. So correct answer is cell plate, okay. Now let's move to the second question. Here you have to fill in this blank. Dash is characterized by all the chromosomes coming to lie at the equator. So means they are talking about metaphase. At the equator, which one chromatid connected by its kinetochore to the spindle fibers from one pole and its sister chromatid connected by its kinetochore to spindle fiber from opposite poles. So at metaphase what happens? This is the metaphase plate. Okay, this is the metaphase plate. Chromosomes are aligned here. Okay, chromosomes are aligned at the metaphase plate here also, and spindle fibers are attached to the kinetochore. Alright, so what should be the correct answer? Correct answer is metaphase. Okay, now next let's talk about the next question, which is the synaptonymal complex appears at which stage? Okay, synaptonymal complex, we know it is a protein structure, or you can say it is a protein lattice that holds the homologous chromosome together. Hold the two homologous chromosome together, and this is seen during the zygotine stage. At zygotine, what happens? Synapsis is observed, which is nothing but pairing of the homologous chromosome. Okay, all right. So synaptonymal complex appears between homologous chromosome. This is correct because it is seen after synapsis. What is synapsis? Pairing of homologous chromosome. It is the pairing of homologous chromosome. Okay, it is seen during the zygotine stage. It is composed of DNA and protein. Okay, so what should be the answer? All these points are correct. So answer should be all of the above. So answer is option D, all of the above. Now next question is about the recombination. Means they are talking about meiosis. And specifically within meiosis, they are talking about prophase 1 of meiosis. Okay, prophase 1 of meiosis 1. And within prophase 1, they are talking about pectine. Because during pectine, recombination and crossing over takes place. So recombination involves first, they are saying crossing over. First option is, second option is pectine. It is related to pectine. Spindle formation. And the last option is cytokinesis. So recombination involves, so what happens? After recombination, there is crossing over. Means there is exchange of the genetic material. There is exchange of the genetic material, which is the source of the variation. Alright. So if this is a chromosome, say, suppose, okay. So here, crossing over is taking place at this point. And there is exchange of the genetic material. Okay, so this is recombination and crossing over, which takes place between two non-sister chromatid. Okay, non-sister chromatid. So what is the answer? Answer is recombination involves crossing over. Now next question is, in which stage the chromosome appear thin and long and thread-like? Okay, and the options are zygotine, leptotine, pectine and prophase. Okay, so initially what happens? We have prophase. Okay, prophase 1 they are talking about. So prophase 1 is further divided into leptotine. We are talking about the prophase 1 of meiosis 1, which is the reductional division. Leptotine, okay, then we have zygotine, then we have pectine, then fourth substage is diplotine, and last stage is diakinesis. Alright, so chromosome appear as thin and long thread-like structure. So during leptotine what happens? Chromatin start condensing. Chromatin is condensing and they form thin, long thread-like structure. Zygotine again, chromosome, they pair up, they form synapses. During pectine, crossing over is taking place. Diplotine, chiasmata is seen, and during diakinesis what is observed? Terminalization of chiasmata is observed. So what should be the answer? During leptotine, the chromosome are visible as thin and long thread-like structure due to the chromatin condensation. Alright, so this was the end of today's session. Hope you enjoyed it, and thank you very much for listening.